Introduction to Heredity and Evolution. The living world is composed of two major groups of organisms, namely the prokaryotes and the eukaryotes. Prokaryotes are a group of organisms that lack a nucleus and other membrane-bound cell organelles. Eukaryotes, on the other hand, comprise of cells with a very well-defined nucleus and even show the presence of complex structures enclosed within membranes. The main difference between prokaryotes and eukaryotes is that, in the case of prokaryotes, the nuclear material is not enclosed by a nuclear membrane, but is in direct contact with the cytoplasm. In eukaryotes, the nuclear material or the nucleus is enclosed by a double membrane called the nuclear membrane. The nucleus consists of genetic material which is organized in the form of very fine, thin thread-like fibers called the chromatin fibers. During cell division, the chromatin fibers condense and become thick and ribbon-like and are known as chromosomes. Each chromosome, in its condensed form, consists of two chromatids joined at some point along the length. Chromosomes are formed of DNA, that is, deoxyribonucleic acid. The genetic information is stored in long stretches of DNA in the form of units called as genes. A gene is thus a specific length of a DNA chain which holds the information to control a specific characteristic of an organism. For example, features such as color of hair, rolling or folding of tongue in human beings, etc. There are thousands of genes on a chromosome which control various characteristics of an organism. Each gene is assigned a specific location on the chromosome, which eventually determines the function of that gene. Now let us understand the mechanism of gene function with the help of an activity. Have you ever observed rose plants growing together in a garden? Do they appear similar or do they look very different? Well, when we observe the rose plants in a garden, all of them appear similar. This is because each of these plants is propagated through cuttings from the same parent plant. Similarly, all sugarcane plants growing together in a sugarcane field will also be similar. Plant cutting, also known as striking, is a mode of vegetatively propagating plants through the method of asexual reproduction. The rose plant and the sugarcane plant both can be propagated through stem cuttings. In the case of rose, we take cuttings from the tip of the stem of a mature mother plant, while in the case of sugarcane, we take cuttings of comparatively smaller pieces of stem. The rose cuttings are planted vertically, while the sugarcane cuttings are planted horizontally. Due to the similarity in their method of propagation, these plants do not show much variation in their growth pattern. Now let us study one more example. Each individual is different from the other with respect to characters such as height, weight, body stature, color of hair, etc. They differ even in terms of their daily habits. From these examples, we understand that asexual reproduction results in small amount of variations as compared to sexual reproduction. Why do these variations occur? Let us understand this further. Accumulation of variation during reproduction. In case of humans, the sexes are separate. Human beings reproduce through sexual reproduction. A haploid male gamete fuses with a haploid female gamete to form a diploid zygote with two sets of chromosomes. The zygote formed after fertilization receives one set of chromosomes from the mother and the other set from the father. Thus, there is a recombination of genetic material which ultimately results in variation. A child has certain features similar to that of the mother and certain features similar to that of the father. The offspring thus closely resemble their parents. Do you know how variations present in the parents are passed on to their children? Let us explore this further. The process of reproduction gives rise to new individuals. The offspring so produced are similar to their parents in many essential characters. However, they do show some minor differences. These differences in the characters of the individuals of a species are called variations. The variations produced in the organisms get accumulated. They are passed on from the parents to their offspring, that is, from one generation to the other. This phenomenon is known as inheritance, and the variations are called inherited variations. The members of the successive generation inherit the characters of the previous generation. The significance of a variation shows up only if it continues to be inherited by the offspring of several generations. The inheritance thus essentially provides the general basic pattern of the body with some minor differences for the next generation. When the individuals of the new generation reproduce, their offspring will have the differences that are inherited from the previous generation as well as certain newly created differences. These differences are the variations produced by the reproductive process. Let us understand this concept with the help of an example. Look at the figure carefully. What do you observe? The figure illustrates an example of a single organism undergoing reproduction as seen in asexual mode of reproduction. The parent organism reproduces and gives rise to two new individuals. These two individuals, although similar in body structure with the parent organism, do have some minor differences. Further, in the next generation, the new offspring reproduce individually to produce two new organisms each. These two organisms also resemble their respective parents, but do have certain minor differences. Some of these differences are unique, while some of them are inherited from their parents. Thus, when we observe all 7 organisms together, starting from the original parent, the two offspring and their offspring, we notice that they are all similar in body structure, but do show certain minor differences. Do you know why in asexual reproduction the individuals have only a few differences between them as compared to sexual reproduction? Well, in asexual reproduction, the individuals produced are quite similar to each other and exhibit minor differences among themselves. This is because of small inaccuracies involved during the process of DNA copying. In this way, diversity is created in the subsequent generations. This is observed in all organisms which reproduce through asexual methods. For example, bacteria and eukaryotic algae. In contrast to these, sexual reproduction involves fusion of two different germ cells. Thus, greater variations and hence greater diversities will be generated. Heredity. Inherited traits. Passing on or transmission of physical or mental traits from the parents to the offspring is known as heredity or inheritance. Why does a rat give birth to a rat only and not a cat? This is because the organisms reproduce their own kind and transfer their characters from one generation to the other. That is why a rat will produce young rats similar to itself. The new offspring will exhibit the same body design with a few variations due to some minor differences in them. Living organisms follow the universal law of like begets like. It means that the offspring of dogs will be dogs, pigeons will produce pigeons, and humans will beget humans only. We have learned that organisms reproduce and produce their own kind. But the offsprings produced do not look exactly like parents, and thus it can be said that human population shows a great variation. Let us perform an activity to understand this. Activity: observe the years of all the students of your class. Prepare a list of students having free earlobes and those having attached earlobes. Calculate the percentage of students falling in each category. What is your observation? Well, the percentage of students with free earlobes and those with attached earlobes is 40% and 60% respectively. Now, if you were to observe the earlobes of the parents of these students, you will observe that their pattern of earlobes is the same as that of their children. This character is thus inherited from the parents. Such characteristic features which are inherited from one generation to the other are called traits. These traits are passed on from one generation to the other. Let us now proceed to understand the rules for the inheritance of these traits. Mendel's rules of inheritance. Inheritance is the transfer of hereditary characters or traits from parents to the offspring. The offspring resemble their parents but show certain minor differences. These similarities and differences are because of heredity. The credit for the concept of heredity goes to Sir Gregor Johann Mendel, who was the first person to carry out work on inheritance. Mendel was not only a priest in a monastery but also a naturalist. He conducted several experiments in the monastery garden to study transmission of certain traits in pea plants. He is now recognized as the father of genetics due to his remarkable contribution to the field. Several other scientists had studied the inheritance of traits. However, Mendel blended his knowledge of science and mathematics and was the first person to give the hypothesis of the existence of a factor called the Mendelian factor to explain continuity and variation. This factor is known as gene. He conducted experiments on pea plants and studied the inheritance of certain traits. He concluded that the inheritance of these traits follows a particular pattern. The significance of Mendel's work was not recognized until the turn of the 20th century. In 1900, scientists rediscovered Mendel's work and its importance in the field of genetics. His work was thus appreciated by the society after his death. Based on his work, Mendel put forth certain laws which later became famous as the laws of inheritance. The laws formulated by Mendel constitute the foundation of the modern science of genetics. Let us now begin our study of Mendel's laws with the help of experiments involving the breeding of native pea plants. Mendel's law for inheritance of traits. The laws of inheritance of traits in human beings are related to the fact that both the father and the mother contribute practically equal amount of genetic material to the child. This implies that each trait can be influenced by paternal as well as maternal DNA. If the contribution of both the father and the mother is equal, then for each trait there will be two alternate forms for each character, producing different effects in the child. These alternate forms of a gene are called alleles. Mendel thought on similar lines and conducted experiments on the pea plant that reproduces through sexual reproduction. Based on his observations and results, Mendel formulated the laws of inheritance. Mendel performed his experiments over a century ago, but the results and findings are still very much relevant to modern genetics. It is very interesting to study these experiments. Do you know which plant was selected by Mendel for his work? Mendel selected plants of Pisum sativum, commonly known as garden pea, for his experiments. Mendel chose pea plants because they exhibited clear-cut differences in certain characters which were easy to tell apart. The pea plant has several distinct varieties, each with visible contrasting characters that appear on separate chromosomes. For example, height of plant, coloration of flower, coloration of seed, etc. Let us study Mendel's experiments involving a single pair of contrasting characters in detail. During the course of his experiments, Mendel conducted many crosses involving plants with a single pair of visible contrasting characters. For example, tall and dwarf plants, red and white colored flowers, round and wrinkled seeds, green and yellow colored seeds, etc. are some of the characters. Such a cross, which involves only a single pair of contrasting characters, is called a monohybrid cross. Let us understand this cross with the help of an example. Mendel selected one pea plant bearing red flowers and another pea plant bearing white flowers as parents to perform a monohybrid cross. These plants are considered to be of parental generation or P1 generation. The terms like dominant and recessive were used by Mendel for red and white flowers respectively. Do you know what the words dominant and recessive imply in genetics? A dominant character is that character which hides or masks the expression of its corresponding character, which then becomes recessive. From a single pair of contrasting characters, one will be dominant and the other will be recessive. The red flowering varieties of pea plants were called dominant because cross-pollination of red flowering plants with white flowering plants resulted in offspring with red flowers in the successive generation. The offspring did not exhibit a single white colored flower. The dominant red color did not allow the expression of the recessive white color. Hence, white flowering plants were called recessive. These findings helped Mendel to formulate one important law of inheritance called the law of dominance. It states that in every pair of contrasting characters or alleles, one is more frequently expressed than the other. This allele is referred to as the dominant allele, and the other, which is less likely to be expressed, is known as recessive allele. Likewise, Mendel studied 7 pairs of contrasting characters. Let us discuss about these characteristics. When we consider the shape of a seed, round shape is a dominant trait, while wrinkled shape is a recessive trait. With respect to seed color, yellow color of seed is dominant and green color is recessive. When we consider the color of the flowers, violet or red color is a dominant trait and white color is a recessive trait. With respect to fruit or pod shape, inflated shape of pod is a dominant trait, while constricted shape of pod is a recessive trait. In terms of pod color, green color is dominant and the yellow color of pod is recessive. With respect to position of the flower, axial position is a dominant trait, while terminal position of a flower is a recessive trait. When we consider the height of stem, tallness is a dominant trait, while dwarfness is a recessive trait. Mendel's experiments involved only pea plants with dominant and recessive traits. Through his experiments on pea plants, Mendel deduced that the factors or genes responsible for a particular character were present in pairs, one as dominant and the other as recessive. A particular convention exists for representing the dominant and recessive factors. Dominant genes are represented by capital letters, while the corresponding recessive genes are represented by corresponding small letters of the alphabet. For example, capital R is for red flower and small r is for white flower. Genotype is the genetic constitution of an organism. It is the description of genes present in an organism. Phenotype, on the other hand, refers to the observable characteristic of an organism. For example, red flowering plants. Now, since the genes occur in pairs, the genotype of red flowering plants will be capital R, and that of white flowering plants will be small r, small r. In sexual reproduction, these factors or allele segregate during the process of gamete formation. This results in the formation of two types of gametes, one with capital R factor and the other with small r factor. Based on this observation, Mendel formulated his first law of inheritance called the law of segregation, which states that the alleles that make up a gene separate from each other or segregate during the formation of gametes. The generation of offspring produced by P1 generation of parents is called as the first filial generation or F1 generation, and the new form of offspring resulting from a cross or breeding are called as hybrids. Thus, phenotypically, when a white-flowered parent P1 plant is cross-pollinated with a red-flowered parent plant, the first filial generation F1 generation produces plants with red-colored flowers. Red coloration is dominant and masks the effect of white coloration. That is, when parents having genotype small r are crossed with parents having genotype capital R, two types of gametes are produced, namely capital R and small r. By the combination of gametes, we get F1 generation of plants with genotype capital R, small r. The first filial generation, F1. During his experiments with pea plants, Mendel noticed that all the plants of F1 generation had flowers that were red in color. These red flowering varieties of plants were different from those of P1 generation. From these observations, Mendel concluded that red color was dominant over white. Although the F1 individuals were bearing red-colored flowers, they also contained factors responsible for the development of white color. That is, their genotype was capital R, small r. These F1 plants produced red flowers which were clearly visible. That is, their external appearance or the phenotype was red. Thus, these F1 plants are phenotypically red, but genotypically hybrids with a combination of capital R, small r. In this experimental cross, red-colored F1 plants are similar in their phenotype, but different in their genotype in comparison to the red-colored parental P1 plant. That is, P1 plants are capital R, and F1 plants are capital R, small r. Mendel was curious to find out the result if he crossed two F1 individuals having this genetically hybrid combination. Mendel thus continued his experiments with these F1 plants. These F1 hybrid plants during gamete formation produced two different gametes, namely capital R and small r. He crossed the plants among themselves or carried out self-pollination, that is, capital R small r cross capital R small r. This resulted in a successively new generation different from the previous one, which is called as the second filial generation or F2 generation. The second filial generation, F2. The F2 generation resulted from selfing of F1 plants, showed the presence of both red-colored flowers as well as white-colored flowers. Mendel experimentally observed that out of a total of 929 progeny, about 705 plants bore red flowers, while 224 bore white flowers. If we calculate the ratio of the same, it is approximately 3 parts red is to 1 part white, or simply 3:1. When we calculate the genotypic ratio, it is 1 capital R is to 2 capital R small r is to 1 small r. Thus, phenotypically there are two types of progeny, namely red-flowered plants and white-flowered plants, while genotypically there are three types of progeny, namely F2 pure dominance with constitution capital R and producing red flowers, F2 pure recessive with constitution small r and producing white flowers, F2 hybrids with constitution capital R small r and producing red flowers due to the dominant gene capital R. The F2 pure dominance and pure recessives are both homozygous, showing the presence of two same types of alleles in the zygote after fertilization. The F2 hybrids are, however, heterozygous, showing the presence of two different types of alleles in the zygote after fertilization. Do you know why the phenotypic and the genotypic ratios are different in offspring? The phenotypic ratio is the proportion of detectable characters of an individual. The genotypic ratio is the proportion of genotypes found in individuals after a cross and is based on genetic composition. As there are dominant and recessive genes existing for a particular character, the genetic composition is influenced by the presence of even one dominant gene and produces the effect in the form of expression of the dominant feature. That is why the phenotypic and the genotypic ratios are different in organisms. For example, in a cross of pea plant involving red and white-colored flowers, the phenotypic ratio of F2 generation is 3 red is to 1 white, but the genotypic ratio is 1 capital R is to 2 capital R small r is to 1 small r. Mendel's experiments involved 7 pairs of contrasting characters. Let us study one more cross of pea plants with some other pair of contrasting characters. Mendel's monohybrid cross. Let us consider pea plants having a pair of contrasting characters with respect to the height of the stem, namely tallness and dwarfness. The parental generation P1 generation has the genotype capital T for a pure tall parent plant and small t for a pure short, that is a dwarf plant. Both the parents are homozygous, having one type of gamete each, that is, capital T from tall parent and small t from dwarf parent. These two plants are crossed with one another. The first filial generation, that is F1 generation, has genetic constitution capital T small t. It is a genotypically hybrid and heterozygous plant having two different alleles. Phenotypically, it is tall because the allele or the gene for tallness, that is capital T, is dominant, which masks the effect of its corresponding recessive gene. It is possible to find out whether F1 generation has traits for both tallness and dwarfness by conducting a cross among these individual plants. Hence, F1 plants are self-pollinated to obtain F2 generation, that is, F1 crossed with F1 gives F2. Now each of these parents is heterozygous tall with genotype capital T small t. So we have capital T small t cross capital T small t. The gametes produced from each of the parents are of two types, capital T and small t. The second filial generation, that is F2, has a genotypic ratio of 1 capital T is to 2 capital T small t is to 1 small t small t. As in this case, allele capital T for tallness is dominant, the P plants with genotype capital T small t will be tall. Thus, phenotypic ratio is 3 tall plants is to only 1 dwarf plant. Genotypically, it shows 3 types of plants. 1 capital T which is homozygous tall, 2 capital T small t which is heterozygous tall, and 1 small t which is homozygous dwarf. Thus, the genotypic ratio would be 1 capital T is to 2 capital T small t is to 1 small t. So far, we have studied the experiments of Mendel with a single pair of contrasting characters. But he had also performed experiments involving more than one pair of contrasting characters.
Mendel's dihybrid cross.
Parental generation P1. A cross which involves plants with two pairs of contrasting characters is called a dihybrid cross. For example, if tall plants having round seeds are crossed with short plants having wrinkled seeds, then the cross involved is a dihybrid cross. Let us understand about Mendel's experiment involving a cross of pea plants having round and yellow seeds and those with wrinkled and green seeds. In this cross, the two pairs of characters involved are the seed shape and the seed color. Let us analyze the cross step by step. To begin with a dihybrid cross, Mendel first obtained purebred pea plants with round and yellow seeds and other purebred plants with wrinkled and green seeds. The letter capital R is used to denote round-shaped seed and capital Y to denote yellow-colored seed. Hence, the genotype of the parent plant is capital R, capital R, capital Y, capital Y. The other parent has wrinkled seeds which are green in color. The letter small r denotes wrinkled seed and small y denotes green-colored seed. Hence, the genotype of the parent is small r, small y, small y. The two parent plants are then crossed, that is, capital R, capital Y, crossed with small y, small y. When the gametes of the P1 generation are formed, each pair of alleles segregate independently of the other. Hence, during gamete formation, R, Y plants produce the gametes R, Y. The gametes R, or Y are not produced by them. Similarly, the other parent r, y produces the gametes r, y. The gametes r, or y are not produced by them. In each of the gametes, a pair of alleles is represented by only one of its members. These findings helped Mendel to formulate his second law of inheritance called the law of independent assortment, which states that alleles of different genes assort independently of one another during gamete formation. Thus, in P1 generation, a parent R, Y produces two gametes R, Y and R, Y, and parent r, y produces two gametes r, y and r, y. Let us study the result of this cross in the next generation, that is, the first filial generation F1 generation. First filial generation F1. Mendel had observed that only the dominant character gets expressed in the F1 generation. On the basis of these experiments, Mendel anticipated that the F1 generation plants would show round-shaped and yellow-colored seeds. His prediction was found to be correct, and phenotypically the F1 plants were with round yellow seeds. The genotype of these F1 hybrids was R, Y. The genotype shows R, which is dominant over r with respect to the shape of the seed and results in round seeds. The genotype also contains Y, which is dominant over y with respect to seed color and results in yellow-colored seeds. Yellow color is dominant over green, while round shape is dominant over wrinkled. Thus, round yellow seeds are dominant and mask the expression of wrinkled green seeds in the F1 generation. All these F1 plants are called dihybrids as they have two sets of contrasting characters and have the genotype R, Y. The F1 plants produced four types of gametes during gamete formation. These are r, y, R, Y, r, Y, and R, y respectively. On the other hand, parental plants P1 produced only two types of gametes R, Y, and r, y. The gametes produced by F1 plants that are similar to those produced by P1 plants are called parental combinations. The two other types of gametes, namely r, Y and R, y, produced by the F1 plants during gamete formation are called as recombinations. When the F1 plants are self-pollinated, they produce a new generation of plants known as F2 or second filial generation. The four types of male gametes and the four types of female gametes produce in all 16 different mating combinations. Reginald C. Punnett developed a tabular system of representing these different combinations.
This system was used by Mendel, which helped in easy identification of different genotypes and phenotypes.
This type of Punnett square is also known as checkerboard. The male gametes are arranged on top, while the female gametes are arranged on the side of the checkerboard. Taking into account the combinations of a male with a female gamete, we get the genotype of a hybrid individual in a respective cell of the checkerboard. Thus, 16 different types of F2 individuals can be easily represented with the help of a Punnett square. Second filial generation, F2. The F2 generation obtained by self-pollination of F1 plants exhibits plants with different genotypes. Out of a total of 16 plants, 9 plants are with round and yellow seeds, capital R, capital Y. 3 are with wrinkled and yellow seeds, small r, capital Y. 3 are with round and green seeds, capital R, small y. And 1 is with wrinkled and green seed, small r, small y. Thus, the phenotypic ratio of the F2 plants is 9:3:3:1. Now, let us find out how much we have understood about a dihybrid cross by analyzing one more example of pea plants. Assume that a purebred tall pea plant bearing red flowers is cross-pollinated with a short pea plant bearing white flowers. The cross involves a dihybrid cross with 2 pairs of contrasting characters, namely the height of plants and the color of flowers. One of the parents is tall, bearing red flowers, and the other parent is a dwarf, bearing white flowers. Tall plant and short plant are represented by genotype T and small t respectively. Capital R represents the genotype of red flowered plants and small r represents the white flowered plants. P1 generation of plants is represented by T, R and small t, small r. These 2 parental plants are then cross-pollinated, that is, T, R cross small t, small r, small r. Gametes produced by P1 generation are T, R and small t, small r respectively. F1 generation of plants have genotype T, small t, R, small r. The F1 generation is then self-pollinated, that is T, small t, R, small r, cross T, small t, R, small r. F1 generation of plants produce 4 types of gametes each. T, R, small t, R, T, small r, and small t, small r. Let us now calculate the number of F2 progeny with the help of a Punnett square. The phenotypic ratio obtained from the cross is 9:3:3:1. That is, 9 tall red plants is to 3 tall white plants is to 3 short red plants is to 1 short white plant. The genotypic ratio obtained is 1:2:1:4:2:1:2:1. 1 capital T, R, 2 capital T, small t, R, 2 capital T, R, small r. 1 small t, R, 4 capital T, R, small r. 2 small t, R, small r. 1 capital T, small r, 2 capital T, small r, small r. 1 small t, small r, small r. Mendel was the first to clearly put forward the idea that the hereditary units are present as definite particles and are referred to as factors. These were later known as genes, which are functional segments of DNA.
Expression of traits.
You all are aware of DNA. DNA is an abbreviation for deoxyribonucleic acid, which is one of the nucleic acids present in the nucleus of all eukaryotic organisms. The nucleus contains chromatin fibers composed of DNA and proteins. DNA molecules transfer the hereditary information to the next generation. DNA is a highly complex molecule having spirally coiled double helical structure which appears like a ladder. The most widely accepted model of the structure of DNA molecule was proposed by Watson and Crick in 1953. They received the Nobel Prize for this discovery in medicine in 1962. Let us now study the expression of particular traits and the mechanism of heredity. The genetic material present in almost all living organisms is DNA. It is the source of information for protein synthesis required by the cell. A gene is the section of DNA on a chromosome which codes for the formation of a protein controlling a specific characteristic or trait of an organism. We will understand this with the help of a trait, for example, tallness in plants. The plants contain hormones which control the growth of plants. The increase in height of plants depends on the amount of growth hormones produced by the plant. The amount of hormone produced by a plant, in turn, depends on the efficiency of the enzyme. Efficient enzymes allow hormones to be produced in greater quantity. Hence, the plants gain height and they appear tall. Now, if the gene coding for the enzyme is expressed to a greater extent, then the enzyme produced will be in larger amounts. The greater the amount of enzyme, the more efficient will be the reaction producing the hormone. Eventually, more the hormones, more will be its effect in the growth of plants into tall trees. If the enzymes involved in the process of hormone production are less efficient, then less quantity of growth hormone will be produced. That is why the plants do not grow and appear stunted. The Mendelian experiments suggest that during sexual reproduction, fusion of gametes takes place. Each parent of the parental generation P1 generation contributes equally a copy of the same gene to the DNA of the progeny. Each pea plant thus has two sets of every gene called alleles, one received from the male parent and the other from the female parent. But each gamete, however, has only one gene set. Let us now understand the mechanism by which the germ cells make a single set of genes from the two sets received from the parents. In a dihybrid cross, if the progeny inherited only one gene set from each parent, then two traits in the Mendel's experiment, namely shape and color of the seed, would coexist. For example, capital R and capital Y would be joined and could not be inherited independently. But this does not happen, as the DNA in the nucleus is not present as a single long thread, but as separate independent pieces called chromosomes. In human beings, there are 46 chromosomes or 23 pairs of chromosomes, one from each parent. During gamete formation, the gamete takes one chromosome from each pair. When two such gametes having single set of genes combine together, they restore the normal diploid number of chromosomes in the progeny. This is the mechanism of inheritance of traits. The results of Mendel's experiments can be explained by this mechanism. In higher organisms, individuals of both male and female sex are involved during sexual reproduction to produce a new individual. How is the sex of the new individual determined? Let us study this.
Sex determination.
In higher organisms, sexes are separate. Male and female sexes are involved in the sexual reproduction. There are various factors which contribute to the determination of sex of an individual. Some environmental parameters besides genetic factors also influence this determination. Let us understand these factors of sex determination. In most of the organisms, genetic or chromosomal mechanisms are mainly responsible for the determination of sex of an individual. For example, in some animals, the temperature at which the fertilized eggs are maintained determines the sex of the offspring. Do human beings also show a similar mechanism of sex determination? Let us explore this further. How is sex determined in human beings? The sex of an individual depends on the genes inherited from its parents. An individual usually inherits a similar set of genes from each of the parents during gamete formation. A certain species of organisms has a specific number of chromosome pairs. Out of these pairs, one pair decides the sex of the individual. This pair of chromosomes is referred to as sex chromosomes. For example, human beings have 23 pairs of chromosomes. In human beings, out of these 23 pairs, 22 pairs are autosomes, which perform functions apart from sex determination, and the last one pair is the sex chromosome, which determines the sex of an individual. In human males, two different types of chromosomes are present in the pair of sex chromosomes. One is longer and the other is shorter. The longer chromosome is the capital X chromosome, and the shorter one is the capital Y chromosome. In human females, the last one pair of sex chromosome shows two similar chromosomes, that is, two long capital X chromosomes. Thus, the last chromosome pair of the male parent has capital X, capital Y, and the female parent has capital X as its genetic constitution. The male parent produces X-bearing and Y-bearing gametes or sperms, while the female produces only X-bearing gametes or eggs. During reproduction, combination of X-bearing gamete from males with one X-bearing gamete from females results in offspring with the genetic constitution XX. In the same way, combination of Y chromosome from males with one X chromosome from females results in an offspring with the genetic constitution XY. Similarly, the other two gametes also result in two new offspring with genetic constitution as XX or XY. Thus, the chance of getting a female child is 50%. That is, 2 out of 4 offspring will be females. Similarly, the chances of getting a male child is also 50%. That is, 2 out of 4 offspring will be males. From this, it is clear that all children will inherit an X chromosome from their mother, irrespective of whether they are boys or girls. This is because the XX genetic combination of a female parent produces only one type of gamete, that with a single X chromosome. Do you know why in human beings the gametes from the male parent decide the sex of the baby? Let us understand this. A male parent has XY sex chromosomes. This parent produces two types of gametes, one with an X and the other with a Y chromosome. A female parent has XX sex chromosomes. Hence, this parent will always produce two similar types of gametes, those with a single X chromosome. Thus, an X chromosome inherited from the mother will always be present in the genetic constitution of the offspring. Thus, it is the type of male gamete involved during fusion which will decide the sex of the offspring. If it is an X chromosome, then the offspring will be a daughter with genetic constitution XX, and if it is a Y chromosome, then the offspring will be a son with XY sex chromosomes. Thus, the sex of a baby is determined by the type of chromosome they inherit from their male parent.
Evolution. An illustration of natural selection.
Charles Robert Darwin was an English naturalist who firmly established the theory of natural selection. He was a Fellow of the Royal Society. He established the fact that all species of life have descended from common ancestors. He proposed that the new species came into existence by a process called natural selection. As a result of careful study and observations of many years, he proposed his own theory of evolution and published it in a book called The Origin of Species in the year 1859. The scientific community began to accept evolution as a fact, and natural selection was considered to be the basic mechanism of evolution. The Darwin's theory is a unifying theory of life science and explains diversity of life. Let us now study Darwin's theory of natural selection. Darwin's theory of natural selection. Darwin assimilated and analyzed the observations of his data and suggested that only those who are fit for the changing environment have the right to survive. The organisms which are unfit are eliminated and ultimately die. The fit species reproduce. The essential variations in the characteristics are passed on from one generation to the next. These variations make them fit to survive in the constantly changing environment. The process of selection of characteristics that contributes to the fitness of survival of organisms was called natural selection by Darwin. There are differences observed even in individuals of the same species. Some individuals have certain factors or traits in more amounts than other individuals. If these factors are essential for the survival of that organism, then those possessing them can survive, while those which lack these necessary factors die eventually in the struggle for existence. Again, there is a possibility that the beneficial or useful factors appear only in a few individuals of the next generation. These few individuals will ultimately survive in the long run. This process continues with the elimination of some unfit organisms generation after generation. So, after several generations, the number of individuals better adapted to their surroundings with all the essential factors will always be more than the previous generation. This process is referred to as natural selection. The individuals having better characteristics suited for adaptation will survive. The desired characters which are transmitted to the offspring are those which are selected by nature. The selection by nature is not deliberate but natural and depends on the ability of successful adaptation for growth and reproduction in a given environment. Let us understand natural selection with the help of an example of industrial melanism as observed in moths in England. Moths basically serve as food for birds. Generally, moths are found in two different colors, black or melanized moths and grey or light-colored moths. These moths live on the tree trunks. The tree trunks in the Birmingham area had the growth of algae and fungi, and thus the bark appeared grey in color. The grey or light-colored moths were better protected because the background was light and the organisms were camouflaged and were not falling an easy prey to the birds. The black moths, however, were easily noticed by the birds and they were consumed by them as food. Their number was less due to predation as compared to light-colored moths. Later, at the beginning of the 20th century, due to rapid industrialization events, there was a steep rise in the level of pollution. Because of the smoke and deposition of soot, the light background eventually became dark. Due to increased level of pollution, growth of algae and fungi on the tree barks was stunted. Now, the dark moths were protected while the light-colored moths were easily noticed and fell prey to the predators. In the area surrounding Birmingham nowadays, we often notice black moths in greater numbers than the light ones. Thus, natural selection helps the well-adapted individuals and organisms to survive. The theory proposed by Darwin helped to explain the developmental process of living organisms. The theory of natural selection, however, did not explain the mechanism by which the living organisms acquired the adaptation factors.
Let us study inherited and acquired traits. Inherited and acquired traits. Inherited traits. The traits or characteristics of an organism which occur due to a change in its genes or DNA are called inherited traits. They are passed on from one generation to another. Let us understand this concept with the help of an example. Assume that there is a population of red beetles in the green bushes. Suppose a color variation arises during the process of reproduction, as a result of which one of the beetles produced is green in color. This change in color has been brought about by the change in the genes of the reproductive cells of the beetle. The green color of the beetle is thus an inherited trait, which can be passed on to the next generations. There will definitely be one red progeny in the successive generations of the beetles. The change in the color of the beetles from red to green is a result of evolution, since it helps in the survival of the beetle within the green bushes. Let us now understand the concept of acquired traits. Acquired traits. A trait or characteristic which develops in response to the environment and cannot be inherited is called an acquired trait. Let us understand this concept with the help of an example. Assume that a beetle does not obtain sufficient food due to unfavorable conditions. As a result, due to starvation, its weight may get reduced. There is a change in the normal body cells of the beetle, but no change occurs in the genes present in the reproductive cells of the beetles. Since there is no change in the genetic composition of the beetle, this trait cannot be inherited by its offspring. Thus, low weight is an acquired trait of the beetle. It will not be passed on to the future generations. Further, if some generations of the beetles are low in body weight, it is not because the trait has been passed on to these generations. It would be purely due to the absence of sufficient food. Whenever sufficient food is available, the trait of low weight would disappear and the beetles would become healthy again. Origin of life on Earth.
Life exists everywhere, from the equatorial region to the extreme poles of the Earth. The organisms are omnipresent, that is, found everywhere, in the air, soil, water, sea, wood, and also the rocks. Man, due to his curious nature, has made several attempts to understand the origin of life on this planet and the great diversity of plants and animals which came into existence. Many theories have been proposed by scientists to explain the origin of life and the diversity of organisms on the earth.
In 1929, J.B.S. Haldane, a British scientist, proposed that the primitive sea served as a vast chemical laboratory powered by solar energy. The atmosphere was oxygen-free, that is, reducing in nature. It was rich in gases like carbon dioxide, hydrogen sulfide, sulfur dioxide, and ammonia. The combination of these gases in the presence of ultraviolet radiation gave rise to a host of organic compounds. The sea became a hot, dilute soup containing large populations of organic molecules. How did these complex organic molecules arise? In 1953, Stanley L. Miller and Harold C. Urey provided the evidence for the existence of organic molecules. Miller took molecules which were believed to be present in the atmosphere of the early Earth and put them into a closed system. The gases they used were methane, CH4, ammonia, NH3, hydrogen, H2, and water, H2O. They maintained the system at a temperature just below 100 degrees Celsius. They ran a continuous electric current through the system to simulate lightning storms. At the end of 1 week, Miller observed that as much as 10 to 15% of the carbon was now in the form of organic compounds. 2% of the carbon had formed some of the amino acids which were used to make proteins. Miller's experiment showed that the organic compounds such as amino acids, which are essential to cellular life, could be made easily under the conditions that scientists believed to be present on the early Earth. But can life arise afresh on Earth even now? No. The primitive environment was reducing in nature, whereas the present environment contains oxygen, that is, it is oxidizing in nature. The presence of oxygen would prevent the formation of organic compounds. This enormous finding inspired a multitude of further experiments. Speciation. All living organisms, including plants and animals, exist in the form of different species. A species is a population of organisms consisting of similar individuals which can breed together and produce fertile offspring. In case of plants, there is a vast diversity observed with respect to the shape of leaves, color of flowers, etc. In spite of this diversity, different plants, for example, wheat, paddy, sunflower, lotus, etc., are considered to be the species of plant kingdom. In case of human beings, although there is a diversity observed with respect to body features such as height, color, etc., all human beings are said to belong to the same species of Homo sapiens. The process by which a new species develops from the existing species is known as speciation. The biologist Orator F. Cook coined the term speciation. Let us now proceed to understand how new species are formed from the existing species. Consider an example of a beetle population feeding on the green bushes spread across the mountain range. A few of these beetles would feed on the bushes in the nearby areas, while some adventurous beetle might travel far off. The population of beetles may get divided into two subpopulations due to geographical barriers such as mountain range, river, ocean, desert, etc. This is called geographical isolation. The two subpopulations are subjected to different selection pressures due to the difference in the conditions of the two areas. Different alleles will be selected and genetic differences will gradually accumulate within the populations. Over a period of time, enough genetic differences will occur, as a result of which the two subpopulations can no longer interbreed. They will emerge out as different species. During the process of evolution, some individuals of a generation may leave behind a few descendants consisting of a set of particular alleles. This results in a drastic change in the frequencies of alleles merely due to chance event. This is what we call as genetic drift. Genetic drift is an important factor that contributes to the formation of new species. Another important factor which leads to the formation of new species is the process of natural selection. Natural selection is the process of selection of characteristics that contribute to the fitness of survival of organisms. It is a key mechanism of evolution. The genetic variation within a population of organisms may cause some individuals to survive and reproduce more successfully than others. Over a period of time, the individuals of the isolated population become completely different from each other. No reproduction is possible amongst these individuals even if they happen to meet again. This incapability of breeding leads to the formation of two new species from a single existing species. This is known as speciation. Out of these several factors, geographical isolation is the major factor in the speciation of sexually reproducing animals, since it interrupts the flow of genes. However, it may not have any effect in case of self-pollinating or asexually reproducing plant species, as they do not depend on the other organisms for reproduction. Evolution and classification. All living things are grouped into different categories on the basis of their body design. As time passes, certain characteristics begin to influence the body design. Once this body design comes into existence, changes begin to occur in it, which in turn bring out new changes. This means that the characteristics which came into existence earlier are likely to be more primitive than those which arise later. In other words, classification is closely related to evolution. Most of the organisms have arisen by an accumulation of changes in their body design. The organisms which possess these favorable changes survive better in the long run. In 1859, Charles Darwin first described this concept of evolution in his book, The Origin of Species. There are certain groups of organisms that have ancient body designs and are referred to as primitive or lower organisms, while some organisms have acquired their body designs relatively recently and are called advanced or higher organisms. However, these terms do not clearly reflect the evolution of these organisms, and hence we often refer to them as older and younger organisms respectively. there is a strong possibility that complexity within the organisms increases with increase in the evolutionary time. Hence, we can say that older organisms are simpler, while younger organisms are more complex. Depending on these different characteristics, a hierarchy is developed that allows us to make classification groups. Thus, different groups can be developed as a small group of species with advanced ancestors, then supergroups of these groups with more distant common ancestors, and so on. If in the same way the hierarchy is followed, we can reach to the beginning of the evolutionary time, thereby proving that at some point in the history of the Earth, non-living material might have given rise to life. Tracing evolutionary relationships. Several biological studies have suggested that right from their origin, living organisms have been undergoing changes in their organization to evolve into new forms. A number of common features of different organisms provide an evidence in favor of evolution. The more characteristics two species have in common, the more closely related they are. The more closely they are related, the more recently they will have had a common ancestor. The characteristics that accumulate in a particular generation can be used to trace the evolutionary history. Molecular phylogeny is the technique used to estimate and analyze evolutionary relationships at the DNA level. The idea is that organisms which are more distantly related will accumulate a greater number of differences in their DNA. One of the most important evidence of the evolutionary relationship is provided by the anatomy of certain organisms.
Anatomical evidence. Anatomical evidence of evolution is usually reflected in the form of structures which appear quite similar in their organization. For example, a human arm, a bull's leg, a bat's wing, and a whale's fin are different body organs which are not similar to one another. Their functions are also quite different. The human arm is used for making different movements such as picking up a book, throwing a ball, etc. A bull uses his leg for activities such as walking, running, etc. A bat uses its wings for flying, while a whale uses its fin for swimming. Due to the differences in their functions, they show a difference in their external structural morphology. But when we observe the internal structure of bones of these different organs, we can see certain similarities. This similarity exists between the arrangement of bones and the joints. Such similarities suggest that these animals are the descendants of a common ancestor. Different organisms have different organs which are fundamentally same in structure but are modified to perform different functions in different organisms are called homologous organs. For example, the 4 limbs of man are adapted for handling, while the 4 limbs of bat are adapted for flying, and those of aquatic animals, whales, are adapted for swimming. All these organs of different animals show the same underlying composition of the skeleton. In plants, the scales of ovulate or female cone of pine correspond with the carpels of a flower of higher plants. The scales of the staminate or male cone of pines correspond with the stamens of a flower. The homologous organs show similarities due to the fact that the process of evolution results in modification of the existing structures. These modified structures perform new modified functions.
Fossils.
Have you ever heard of fossils? Do you know what are they? Let us try to find out. Fossils are the preserved remains or traces of animals, plants, and other organisms from the remote past. Fossils are formed by a continuous process of burying and decomposition over a period of time. The hard parts of the body like skeleton, shell, teeth, and occasionally the entire animal are found embedded in the sediments. These sediments form the rocks. Paleontology is a branch of science which involves the study of fossils of plants and animals found in the sedimentary rocks. The process of fossilization is a long-term process. As rocks exist in the deeper layers of land, the land needs to be excavated in order to study the fossils. Fossils can be collected from different layers of rocks found at different depths. The occurrence of these fossils and their systematic study indicated that the fossils of invertebrates are situated at the deepest level. A layer above them were found the prehistoric fish-like animals, above which were the amphibians. The reptiles and the birds as well as mammals were found to be in the uppermost layer. This record suggests that the simple life forms are found in the lower or the old strata, while the complex forms appear in the upper or recent strata of the earth. Do you know how paleontological evidence suggests that invertebrates came into existence before the vertebrates? The fossils of invertebrate animals are found in the deepest layers of the rocks, whereas fossils of vertebrates, namely the birds and mammals, are found in the recent layers of rocks. This paleontological evidence suggests that the invertebrates came into existence before the vertebrates and reflects the order in which these animals appeared on the earth.
There are two different methods by which we can estimate the age of fossils. If we dig into the earth, we find that the fossils closer to the surface are more recent as compared to the fossils found in the deeper layers. The age of the fossils can be estimated by carbon dating method. When living organisms change into fossils, their C14 carbon-14 radioactive decreases slowly. Thus, the age of fossil can be determined with the help of the C14 radioactive. The age of ammonite, trilobite, and dinosaur is determined by this process. Evolution by stages. There are enormous number of plants and animals found on the planet Earth. How has this great variety of organisms evolved? Well, the organisms which existed on the Earth initially were quite different from the ones present now. They have modified themselves in response to their environment. These changes have occurred gradually in stages and have resulted into the evolution of a new species. The occurrence of different stages of evolution in a species is not because of a single DNA change. Let us understand this concept with the help of an example of evolution of eyes and that of feathers in the organisms. Evolution of eyes. The eye is an example of a homologous organ. It is a complex organ. It has been created in stages over generations. The primitive organisms which existed on the earth were slow-moving and smaller in size. They did not require any specialized organ for observing any object. Consider the example of flatworms. Flatworms are small invertebrates. The eyes of flatworms are simple and exist in the form of eye spots. Eye spots are light-sensitive cells which can detect light. As evolution progressed, comparatively large and mobile organisms were evolved. Most of them were predators and required a better vision for the purpose of predation. Hence, from the basic design of eyes, more and more complex forms were evolved. Insects, octopus, invertebrates all have eyes. However, the structure of eyes differs in each of these organisms. This suggests that they have separate evolutionary origin. The evolution of eyes, therefore, is an example of evolution by stages. Evolution of feathers. You must have seen a bird making use of its feathers for the purpose of flying. But the feathers were not evolved for the purpose of flight. They were evolved as a means of providing insulation to their bodies in cold weather. Scientists have found out the fossils of some dinosaurs that had feathers for insulation against cold weather. Since the dinosaurs were reptiles, it implies that birds have evolved from reptiles. Thus, it can be said that a property which is useful for few species at a period of time can be used later for quite a different function in different species. Does the influence of man have any effect on the evolution of a species? Yes. Artificial selection is the process in which human preferences have a significant effect on the evolution of a particular species. For example, as observed in the case of wild cabbage. Humans cultivate wild cabbage as a source of food and have produced different varieties from it by means of artificial selection. Common vegetables such as cabbage, kale, broccoli, cauliflower, kohlrabi are the descendants of wild cabbage. Cabbage was produced by selecting for a short petiole. Broccoli was developed by selecting the arrested flower development of wild cabbage. Another variety, known as cauliflower, was developed by selecting sterile flowers in the wild cabbage plant. Selection of the swollen parts of wild cabbage led to the evolution of yet another variety known as kohlrabi. Similarly, kale was developed by selecting large leaves. Artificial selection has helped in creating diversity in plants and animals. In agriculture, superior strains of corn, wheat, and soya bean have resulted from the process of artificial selection. Evolution by time. Evolution is the change over time in one or more inherited characteristics. These include anatomical, biochemical, or behavioral characteristics found in populations of organisms. It has resulted in the generation of new varieties of species. So, can we say that evolution has resulted in the progress of mankind? No. In fact, there is no real progress in the concept of evolution. evolution results in the production of diverse life forms subjected to environmental selection. The only progress that has occurred due to evolution is the emergence of more and more complex body designs of organisms. Let us understand this. During the process of speciation, new species are formed from the already existing species. This does not mean that old species are eliminated from the earth. The selection is purely based on the environmental pressure. The newly formed species may or may not be superior as compared to the older ones. It is just that the phenomenon of genetic drift and natural selection have favored the production of these species. They have a different body design and are unable to interbreed with the older population. Again, it is not that the body designs of older organisms were inefficient. This fact is relevant since even today many older and simpler forms of organisms still survive on the earth. For example, bacteria are still found to inhabit some of the most extreme and harsh habitats such as hot springs, deep-sea thermal vents, and the ice in Antarctica, where most of the other organisms cannot survive. When we consider the evolutionary history of man, we often say that human beings have evolved from chimpanzees. However, this is not the case. In fact, both chimpanzees and human beings had a common ancestor long time ago. The two offspring of that common ancestor have evolved differently to form the modern-day chimpanzee and human beings. Human evolution. Human evolution has been studied by using some tools like digging earth, time dating, studying fossils, and determining DNA sequence for tracing evolutionary relationship. The future of human is stable, vast, and full of diversities. There is a great diversity of human forms and features across the earth. Man thought for a long time about different races of human. The human race was identified on the commonest way of their skin color, such as yellow, black, white, or brown. It is now known that the human races have not evolved differently. In the recent years, the evidence has become very clear that all the human beings are of single species called Homo sapiens. By research, it has been established that we have come into existence only few thousand years ago. We all have come from Africa. Our genetic footprints can be traced back to our African roots. A couple of 100,000 years ago, some of our ancestors left Africa while others stayed back. Those who left Africa slowly spread over the planet, from Africa to West Asia, Central Asia, Eurasia, South Asia, and East Asia. They migrated down the island of Indonesia and Philippines to Australia and reached America. They went in groups, sometimes separated from each other and mixed with each other, and even moved in and out of Africa. Like all other species on the earth, they had come into being as accident of evolution.